hear me if I stand like this? And I can. Yeah, it's hard to see anyway. Thanks for the possibility to talk here. So it has been a privilege to have been working with the DTRF. I mean, they've supported a lot of our research. And so we work with Xenopus, so there's still patients or patient-related people around. So Xenopus is a frog. So you may wonder why we use this animal, so it's not a mouse. Well, one thing that is very easy to do in this Xenopus is to knock out genes using CRISPR technology. And especially it's very easy to knock out genes. It's not so easy to knock in genes, but knocking out APC, for instance, is very easy to do. The way we do it is in a mosaic fashion. So we inject guide RNAs at a low concentration together with Cas9 in a particular blastomere in the embryo. And then by some chance, some cells, I mean, the cells divide very rapidly in the embryo, but some cells will acquire the B allelic frameshift mutation in APC, and they will further develop into a desmoid tumor, as you can see here and as we have published before. So what we want to do with this now, with this model, is to find dependency genes, I wanted to do, with the idea of if we co-inject a guide RNA for a candidate dependency gene, if it's not a dependency gene in this mosaic setup, we may, you would expect that if you knock out a gene, the tumor can still grow, right? But if we hit the dependency gene, you would expect that the tumor is no longer able to grow, right? Now we're dealing with a mosaic mutant animal, so in reality, even if we co-inject a guide RNA for a dependency gene, these animals will still develop desmoid tumors. However, when we extract the DNA from these tumors and do amplicon sequencing of the targeted region, we find that if it is a dependency gene, we never have B allelic frameshift mutation. So it's always one wild type allele or it is a one in-frame allele. So never B allelic frameshift mutation. So this is the criteria that we use to find these dependency genes. So we found such a gene, EZH2, it's an epigenetic regulator. There's a drug available, FDA approved for treating other types of cancer. And when we add this drug to the water of frogs that have these desmoid tumors, after 3 weeks, we already see shrinkage of the tumor. So the tumor never disappears completely, but shows shrinkage, as we also could see at MRIs. Now, when we put the drug on cell culture, we don't see any particular tumor-specific responses, which make us think that the effect of the drug is indirect. And of course, we got interested in the immune system. As a reminder, so Wnt signaling is associated in other type of cancers with immune cold phenotype. So due to lack of chemoattractants, immune checkpoint, absence of immune checkpoints, or actually presence of immune checkpoints, or presence of immune suppressive cells. But also we have a lot of interest in does the matrix, so desmoid tumors are collagen rich, does this have an effect on the behavior of immune cells, on infiltration, for instance, of immune cells? Now, a disadvantage of working with Xenopus is, of course, tools. We don't have a lot of antibodies that we can use, so we basically have to develop the tools ourselves. And the best way to do this for us is to develop RNA probes. And for this, we started to identify genes that we could use find genes that we could use to identify specific immune cell populations. And we used then, of course, the spleen to make single cell sequencing and find this way in UMAPs, clustering, the classical thing, do then subclusters. And basically, by this way, we could annotate different identities to the different UMAP clouds, let's say, and then allow us to find gene signatures that we can now use for going back to the tumor eventually, and to see are macrophages there, T cells, B cells, and so on and so forth. Now, that is just showing one of the probes that we characterized here in the white pulp of the spleen, the marker for the B cells effect. Now, in this dependency mapping, we also found another gene, and this was a bit surprising, and that gene was CREB3L1. Now, what is CREB3L1? CREB3L1 is a kind of ATF6-like transcription factor, so these are membrane-linked transcription factors. They are resident in the ER, and the idea is that ER stress induces their translocation to the Golgi, where there's a protease that releases the transcription factor from their membrane anchor. And the transcription factor goes to the nucleus, induces genes, and one of the described genes of CREB3L1, also known as Oasis, are actually collagen genes. So this got us really interested in also the idea, what does collagen do? Does it create maybe also a vulnerability? The cells have to deal with a lot of synthesis and secretion to the ER, to the Golgi of collagen. Can be exploited also as a vulnerability if we interfere with the transport. So this is, and also does maybe the collagen matrix create a barrier for the immune system? So these are the questions that we want to address. And when we look back at the tazemetostat treatment, what you can maybe appreciate is that the tumor shrinks, but the appearance of the collagen also changes a bit, from milky kind of appearance to more, let's say, focused, concentration-like. And this is also a question that we would like to answer. Does the EZH2 inhibition actually have an effect on the collagen matrix? Now, in order to do this analysis, we want to look at the tumor on its own. So the frogs are small, they are like this, right? So are the froglets that have developed these desmoid tumors are even smaller. So this allows us to analyze the complete tumor. And there's a lot of nice tools available with light sheet imaging, focal light sheet imaging that we are using here, where we have to clear the tissue, so the tissue becomes completely transparent. We can do staining for the collagen, and this just the method that was used is shown here, was published on the right. Actually, what you see is actually the trunk of an elephant embryo, and that shows this nice collagen patterns. Just to give you an idea of the size that can be used in this light sheet imaging, right? So these are our own images here of a desmoid tumor, and I have to say that stitching needs some improvement, but we need to do the stitching on a stronger computer, in fact. What you see here in red are the blood vessels that are coming up. So because the erythrocytes in amphibia and in birds are nucleated, so you can actually see the blood vessels. But this allows you to get now the complete architecture of the complete tumor, also where it's infiltrating and so on and so forth. And of course, what we want to do is now on this samples also map then the immune cells populations to see how they are localized, how they behave on, for instance, tazemetostat treatment and so on. So something of interest is here, shown here. This is a recent bioarchive, so it's not peer-reviewed, but a paper that shows that this light sheet imaging can also be done on archived material, so also on archived human material, which may be, of course, of interest. So they're showing here, oh, no, it's not shown. I skipped the slide, it turns out. Allows you to do on archived material kind of 3D analysis of relatively small samples, of course, not a 10-centimeter tumor. Then finally, just I would like to this final slide show you some additional Xenopus models that we are generating now, and I'm happy to say that we have now, what we have been working with is Xenopus tropicalis. Why? Because that's a diploid animal, right? And it's a tropical animal, but it takes it's not so easy to work with, so you cannot just distribute it. However, Xenopus laevis, you might be aware, this was distributed to clinics as a pregnancy test in the 50s. So it's a very sturdy animal, very easy to use, but it's an allotetraploid. That means it has 2 alleles of the APC. So that means that we have to actually inject 2 guide RNAs for the 2 alleles, but I'm happy to say that this was not a problem. So we had also desmoid tumors in these bigger Xenopus laevis. And also of interest, maybe it's, we can do this in an albino, or we did it also in an albino wind reporter. So the green that you see there is actually the tumor that you see from the outside when you expose it to UV light. And then finally, we published before that we were able to induce tumors, desmoid tumors, with the S45P mutation by using CRISPR-based editors. But this method, base editors, did not allow us to introduce the other missense mutations. But now we have used CRISPR prime editing, and we were able to induce a T41A. And actually, the same approach will be used and should be useful. Should be possible to use to also have the S45F and the S45B mutations introduced in the animal. And with this, I thank the DTRF, the Desmoid Tumor Foundation of Canada as well, and as well as Desmoid and all other sponsors. And I'm happy to take questions. I'm not going to answer any question.
So I'll ask a question. I was going to ask a question. So this CREB3L1, tell me, so what is the plan for that? If you knock it out, will the collagen stop being excessive? Is it
yeah, you would expect that there would be reduced expression of CREB3L1. The thing is, it's not going to be easy to drug it.
Is there a human equivalent, or is that the human?
That's a gene name. So, and I'm happy to say CD63 is there as well. Collagen 25A1 is there as well.
Oh, collagen 25A. So it's there.
Oh, basically most of the genes are there. Yeah. Yeah.
So how did tazemetostat get tested? What made you take tazemetostat initially? I was kind of curious. What was the rationale for testing tazemetostat?
Because we found EZH2 as a dependency gene. So, and it's the inhibitor of EZH2. So in this case, of course, the drug is only added to the water, just added to the water. So it's taken up by the animal. So it would be more difficult for the FOC 001. So if that's intravenous, we're going to experiment a little bit with some intratumoral injections as well. So maybe that there could be possibilities there.
Does it affect it? Is it an epigenetic modifier? What is the mechanism then? What is the mechanism for tazemetostat in terms of how's it functioning in the model?
That's what we try to understand. Yeah. So we have evidence that it's an indirect effect. So that
yes,
makes
you said
the environment, the tumor microenvironment has the effect on the drug. But it's a rapid response. So it was only 3 weeks when you see already see the shrinkage. All right. Thank you.